And this culture here, this is with a new media here that we're now going to inoculate. Now, do you see how I have the tubes spread out in a V? Okay. Why so?
Keep the lid separated?
Right, to keep them separated, physical distance between them, so that maybe bacteria on the lid, if it was too close, then maybe somebody banged me, then they got jiggled.
Just eliminates the possibility.
Right, eliminating the possibility of contamination through physical distance. Okay. Also, if you notice, my pinky is kind of where the tubes or cultures are sitting on, the tubes, right? The test tubes. Okay. See, my pinky is what the two test tubes are sitting on. And then my thumb here just holds it in place. Right. Why is this important?
So you don't drop it.
Right, so you don't drop it. And what if you get jittery?
Calm down.
Right, just relax. Just relax. You're not getting graded on this, and I'm not going to yell at you, okay? Just relax, be calm. Always be calm. Why? Because the Bunsen burner is also going to be on, right? What if you get all nervous and then your lab coat catches on fire? So just relax, okay? Always think about what we're doing. Before you even do the real thing, sit there and think about what we are really going to do, the steps that you're really going to do. Okay. Now, I'm got my culture set up in the V. I'm ready to proceed. I pick up my inoculating loop. So everyone can see that it's a continuous loop, a complete loop, right? Right? A complete loop, okay? Because why is it important? The liquid from here is going to sit right inside of this loop, right? Just like if you're going to blow bubbles, you know, like that. You have a continuous loop. So when you pick your inoculating loop, make sure it's continuous and a continuous circle, okay? Because that's what the liquid culture is going to sit in. The other thing is, what if your loop was bent? That. Well, bend it back so that it is straight. Okay? Straight. Okay. I'm ready to proceed. I straightened out my loop. I'm ready to take from our mother culture and inoculate our culture that we're going to grow overnight. Okay? First thing I'm going to do is flame and aseptically clean my needle. Okay? So I'm going to hold it at an angle, right, like so, and I'm going to flame it till it's glowing red hot, right? And then the whole tip here is already glowing red hot. I'm going to go further and deeper so that this beginning part can be cleaned and sterilized also. Okay? Sterilizing it. Great. It's really hot right now, and it's best that I don't even touch it. It's too hot. It's going to burn you. So too hot to take bacteria, right? It's going to be burning the bacteria. So the next thing you do is, right. So now we will proceed to open our caps. How? So we hold the loop like this, and we leave a little bit of an inch, right? Inch out there. We don't hold it too close like that, because we might get close and your hand's too close to the culture. You might drop some things, okay? So hold it about there where you have about an inch there, okay? Now, I just flamed it, it's burning hot. In the meantime, what are we going to do? Take off our caps, right? We're going to take first from the mother culture, okay? Take always first from the mother culture. And now the cap rests in your pinky. Okay, you see that? Okay. And then now, with your ring finger, you proceed to take off the other cap. Wait, hold on. Here. Okay. So it's kind of hard. You just got to work with it. Maybe you loosen it beforehand, yeah? Be good. Why don't we just loosen it beforehand? So now we just get up like so. Next thing, we flame, right? Flame. Because just because our bacteria culture, we know this is a bacteria culture we want to inoculate from. Who's to say that from the air, other microbes didn't collect around the rim? So we flame it. Flame the rim. Done. Our needle is still clean. We're now going to take in. Main thing is, you always remember, you can move your wrist and move your arm. Okay? Put it right in front of you. I want you to watch the loop go in there, all the way till you reach kind of like the bottom. And now pull out. Okay. Most importantly, you're not nervous and rattling like that. You're going to contaminate it already. Okay. Other thing is, we put the loop in right in the center of the tube. We never bring it along the edges. Okay. Never push the loop along the edges. With your eyes, watch it and go in directly in the center. Pull it out, and right away, watch, put it in, stir it up a bit, pull it out. Next thing you do, don't worry about the needle. We're worried about this culture. This is the culture that we're inoculating. We don't want contamination in this culture, okay? Because this is the one we're going to observe on Thursday. Flame. And again, cap, first the mother, and then our new culture, and right away, close it. And then, flame the needle. Flame it well. Red hot. Okay. Start again. I forgot to tell you something. When you actually do the real thing, we're all going to practice a dry run. We're all going to do the dry run. But if you do the real thing, now, sometimes our bacteria are going to be clumping on the bottom. So what you want to do before you start is shake it up a bit. Okay? And then proceed to the next steps. Okay. So starting from the beginning. Okay, so you form your V. Very, very calmly, right? We're going to proceed with the next steps. Ready? This is our mother culture that we're going to take from the bacteria and then inoculate our culture. We're going to start by flaming our needle. Flame it well. Okay? And then further and further. Okay? Next thing, make sure the caps are ajar. Take them off. Obviously, this is not an easy kind of thing to do when you're working with two things. All right. I didn't ajar it enough. Unfortunately. So don't touch it when you're really good. What happens if you're losing grip? Put it back on. Okay? That's the best thing. Because it can happen. Okay? So don't worry. You didn't contaminate it. Now lift up. And then lift up. And now you're ready to take from your mother culture. Get a loop full. Pull it out. Next, put it into our culture that we're inoculating. Okay, just to get that water, that inoculum into ours. Pull it out.
Were we supposed to
Flame it first.
before you did?
Right. Start again. Sorry. Starting again from the beginning. We're going to flame the needle. Flame it.
That's after we have to.
Then we're going to take off our lid. Okay. And the mother culture is always on your pinky. So if you forget, mother is always pinky. And our new culture is always held by your ring finger. Now we're going to flame it. All right. Now we're ready to take the inoculum.